Hi, we are the Lethbridge Collegiate iGEM team, and for our project this year, we are designing a CRISPR-Cas13a target-specific system to combat the issue of harmful algal blooms in southern Alberta and beyond.
Cyanobacteria are a natural part of lakes and oceans at nearly undetectable levels. However, their presence has increased to problematic levels in recent years due to increased levels of phosphorus and nitrogen in bodies of water. Our prairies have an especially large blue-green algae problem, as prairie soil naturally contains more phosphorus to begin with, and many prairie lakes have farms nearby. This creates the conditions for algae to thrive. When these algae blooms decay, they consume large amounts of oxygen. This can deplete oxygen levels below the threshold for aquatic organism survival. The current treatment methods have many problems. Treating water with copper sulfate or herbicides also kills algae that are necessary for healthy aquatic ecosystems and does not deal with the large release of microsystems due to the death of cyanobacteria. Trying to regulate the use of fertilizers by everyone is difficult, especially in Alberta, where agriculture is such a prevalent industry. Additionally, while this may lead to fewer problems with cyanoblooms in the future, it will not solve the current problem already present.
To address the widespread harm caused by blue-green algae, or cyanobacteria, we designed Cyanomore. Our proposed synthetic biology solution involves delivering a CRISPR-Cas13a system into cyanobacteria cells. This will be achieved by using a phage-like particle encapsulating our system. Upon being delivered into the cells, our system offers the advantage of being highly specific. CRISPR-Cas13 uses a CRISPR RNA, which is designed to be complementary with a sequence of messenger RNA from Microcystis aeruginosus cyanobacteria. Once the mRNA CRISPR RNA interaction occurs, the Cas13 enzyme will activate and cleave all surrounding target RNA within the cell, resulting in cell death. Our second strategy is to deliver the enzyme MLRA to cyanobacteria cells. MLRA inactivates harmful microcystin toxins contained within cyanobacteria, mitigating their toxic effects in bodies of water. The interactions of the MLRA enzyme and cyanotoxins are being examined by using molecular dynamics computer simulations. Our model will inform our wet lab studies by giving an accurate prediction of the stability and specificity of MLRA. Through our combined approach of targeting both cyanobacteria cells and the toxins they produce, our goal is to reduce the sickness and harm caused by cyanobacteria. Over the past year, we have explored multiple methods in which we could use phage-like particles to deliver our Cas13 system into cyanobacteria. At this time, we have designed a method of packaging the Cas13 system and MLRA inside the MS2 phage-like particle, which would bind to the cell and deliver the cargo inside. This year, a primary focus of our project will be producing our system in the lab. Our experiments will be designed to assemble the phage-like particle, confirm its assembly and encapsulation of Cas13, as well as test the activity and specificity of Cas13.
In the modeling of our system, we have focused on the microcystinase A, or MLRA enzyme, particularly with improving characterization of its catalytic mechanism for inactivation of harmful microcystins. The tools we have used to accomplish this are a combination of homology modeling, molecular dynamics, or MD, docking, and other programs and software. The model will provide a strong basis for our development of a biocontrol platform involving MLRA and inform our design for the best application method in detoxification of bodies of water.
For human practices, we sought the opinions of individuals with experience and expertise related to our project in order to maximize the effectiveness of our approach. During our first interview with Dr. Greg Pyle, a biology professor at the University of Lethbridge, we discussed the importance of minimizing off-target effects. Since then, we have discussed ways to target cyanobacteria without causing harm to other important organisms, including our current idea to utilize the Cas13a system. We proceeded to conduct interviews with 4 other experts in order to learn more about experimental testing ponds and the importance of target specificity. In the next year, we plan to get into contact with potential stakeholders who would be interested in a new method of dealing with cyanobacteria. These 3 stakeholders include government agencies, the agriculture sector, and water activity enthusiasts.
On behalf of the Lethbridge Collegiate iGEM team, we would like to thank our PI, Dr. Angeliki Pantazzi, for her instruction and for providing the modeling team with computational resources, our advisors Marcel Kupansis, Fabian Roden, and Christy Turton, all of our interviewees and collaborators, the Wetmore Computational Group for their guidance in our system modeling, as well as the Diemüller Lab and Dr. Laura Keffer-Wilkes. We would also like to thank our generous sponsors and all of you for listening to our presentation.